All right, we're going to go ahead and get started. Hello, everyone. Thank you for joining us today. My name is Stacy Schultz-Cherry, and I'm the co-editor-in-chief for the Journal of Virology. The American Society for Microbiology publishes Journal of Virology, which is hosting today's webinar, as well as others in what we call our JVI seminar series. For more information on the series and to watch our introductory episode, please visit the Journal of Virology website or scan the QR codes you see on the slide here. Next slide, please. If you're not familiar with the Journal of Virology, we publish preeminent work in virology research. We welcome primary research, mini reviews, and other article formats that interrogate fundamental processes and structures in viruses, the mechanisms by which they interact and evolve with their host and environment, and things like novel methods, therapeutics, and diagnostic strategies. So we really encompass all of virology and welcome all viruses and viral families. Next slide, please. Before I turn things over to our speakers, let me quickly go through the format of our webinar and some housekeeping rules. So as you can see here, the first 35 minutes is really about the science. This includes the speakers introducing themselves and then really walking through their exciting publication. The next 15 minutes, we're going to discuss the paper and really answer questions about the importance of the studies, why should people care, and what do the authors want us to know? And then we'll have questions and answers in the last 10 minutes. Now for some housekeeping rules. The webinar and future seminar series webinars are recorded and they'll be posted on the Journal of Virology website. Our plan is to host these webinars every other month. And at the end, I'll tell you about the next seminar that we'll be doing. In lieu of a certificate of attendance, you will receive an email confirmation for registering for the webinar. This serves as proof of your attendance. And if you have any questions for our speakers, please use the Q&A button at the bottom of your Zoom screen. We are not using the chat function, so please submit all your questions and comments via the Q&A button. So really excited that our webinar today is entitled Targeting Zika, Virus-like Particles, Vaccines with Authentic Quaternary, I can't say that word, Epitopes for Enhanced Protection. Our speakers co-authored the paper that was published in the Journal of Virology in February 2025. If you have not read the paper, highly encourage you to do so. It's an open access paper, so everyone can access it from the JVI website. Next slide, please. And first, I'm going to introduce our moderator for today, Dr. Monique van Oers, and she's the head of the Laboratory of Virology. Monique, at Wageningen University, I'm so sorry, in the Netherlands. She's dedicated her scientific career towards understanding insect-virus interactions with a focus on really functional genomics of large nuclear replicating DNA viruses in insects. She's an expert in the optimization of the baculovirus expression system for vaccine and gene therapy purposes. And in 2019, she received funding from the European Commission to set up and coordinate the European Joint Doctorate Program, Insect Doctors. Training network taught 15 insect pathologists. Really excited to have her. She is one of our outstanding editors at the Journal of Virology, and I am thrilled to turn today's session over to Monique. Over to you.
Yeah, thank you very much, Stacy. Well, I'm not speaking today, so I'm handing over, and I'm delighted to announce my colleague, Professor Gorben Pijlman. He's Professor of Virology and Medical Biotechnology at Wageningen University, so we are colleagues. And Gorben has been serving already for quite a while as editorial board member of Journal of Virology. And he will give a seminar, and the title already has been explained, on Zika virus, like particle vaccine. And it's not only a success story this time, it's really also about the issues, the hurdles on the way to get there. And that's also one of the exciting parts of our work, to make things work and to struggle as long as needed to get things where we want them to be. So without further ado, I hand over to Gorben Pijlman. So Gorben, the floor is yours.
Thanks, Stacy. Thanks, Monique. I'll take over here and talk about the paper on the Zika virus-like particle vaccine that we generated in Wageningen University together with very important collaborators, I have to say. So what you see here is a picture of our campus as you find it in Wageningen, and we're in one of those buildings in the back. Now, today is about Zika, and Zika is an arthropod-borne virus, also abbreviated as arbovirus. And arboviruses are a group of diverse animal and human viruses that have in common that they are transmitted by blood-feeding arthropod vectors. So these critters, they take blood meals, and these can be mosquitoes, ticks, sandflies, and biting midges. And we see a few examples in the slide here. So, for example, in the Netherlands, we have the common house mosquito Culex pipiens as a vector for West Nile and Rift Valley fever. There's tropical mosquitoes that enter our country every now and then. Aedes aegypti is the most important arboviral vector for yellow fever, Zika, dengue, and chick. And there's also other insects like biting midges, which are important transmitters of blue tongue and Smallenberg. And then there's ticks that can transmit tick-borne encephalitis. Now, when I started my group in 2007 at Wageningen University, and I had just spent 4 years as a postdoc in Alex Crommick's group in Brisbane, Australia, where I worked on Kungian virus. Then in 2007, I started up my group here, and this coincided with the first outbreak of chikungunya virus in Italy. So it was the first time that such a tropical virus entered the European mainland, and I thought that would be a nice topic to work on. Now, this outbreak had been ongoing already for a couple of years. It originated in Kenya and then spread to all the islands in the Indian Ocean and made its way into India in a huge outbreak. I wrote lots of grants in my first year, and one was awarded, and that was on the development of a chikungunya virus-like particle vaccine. And we chose to use the baculovirus expression vector system to produce virus-like particles of chikungunya. And I will talk about Zika in a bit. So in this system, we use insect cells that we infect with a recombinant baculovirus that codes for the structural proteins of, in this case, chikungunya virus. And then if you infect insect cells, for example, in a big bioreactor, these cells start producing the VLPs alongside the baculovirus that spreads the infection to other cells. And this is a very powerful system. So in an infected insect cell, then the baculovirus produces a transcript of the chikungunya virus that encodes for the structural protein. So we have the formation of capsid, which makes this capsid shell in the cytoplasm. In the ER, in the meantime, the chikungunya envelope glycoproteins are expressed, are folded, are glycosylated, and are transported through the Golgi system as trimeric spikes, and they end up at the surface of the insect cell. And then the capsid drives the budding by interaction with the glycoproteins, and then these envelope VLPs are released in the culture fluid. And if you purify those from the culture fluid, they look like little donuts in the electron micrograph. And we could produce these VLPs with very high efficiency. So we then teamed up with a former collaborator in Australia, Andreas Zuurbier, and he had just set up a wild-type mouse infection model of arthritic disease in C57 black 6 mice. And if you inject these mice with chikungunya in the foot pad, then about a week later, these feet they swell up. And you can actually measure this foot swelling, and that is a very good measure for chikungunya virus-induced disease, and this model can also be used to test vaccines. So what we did next is we tested our VLPs in this vaccination challenge model, and we vaccinated the mice with different groups of vaccines. So we have a couple of controls. We have a PBS and a GFP group. Then we have our VLP groups in various concentrations, with and without adjuvants. And what you see here in the graph is the readout of vaccinated mice who received a single shot with the vaccines. and then days post-challenge. So at day 0, these mice have been injected with live chikungunya virus, and what you then can see in the control groups is that there is massive foot swelling commencing at day 5, and then within 2 weeks these mice are fine again, they have cleared the virus. Our vaccine groups are flatliners in this assay, meaning that they very well protected these mice against chikungunya disease. Although we needed 1 microgram to vaccinate, if we look at the 0.1 microgram, it is also working, but not good enough. So this was a very successful study, and a similar vaccine, also based on VLPs, is currently authorized by the FDA and EMA in Europe, and should be on the market soon. Now, chikungunya has been circulating in South America since 2013, but Brazil, for example, already suffered from severe dengue outbreaks. Several serotypes of dengue were already circulating in Brazil, and then chikungunya came on top of it using the same Aedes aegypti mosquito. And then in 2015, Zika was introduced in this continent, and that formed another complication, and we'll talk about Zika in this seminar today. So in our laboratory, in the meantime, we had set up a BSL-3 laboratory where we could work with live Zika and chikungunya, and we also had mosquito infection models. And one of the questions that we wanted to ask is what would be the interaction between Zika and chikungunya in the mosquito? And here you see 2 of my former colleagues who did this co-infection study, and they really hypothesized that these 2 viruses may be interacted. But in contrast, what we found is that these viruses, they do not care whether the other virus is there in the mosquito. The Zika virus infection and transmission rate are unchanged when you introduce chikungunya, and also the other way around. Chikungunya infection and transmission rates stay high also in the presence of Zika. What we also saw in this study is that a little percentage of mosquitoes, about 10%, had mosquito saliva that was positive for both Zika and chikungunya, which means in practice that if you're an unlucky person, then you can get 2 viruses in a single mosquito bite. And some colleagues of us in the United States have even shown that you can get 3 viruses in a single bite. So that was an important insight. Now, today is about Zika. Where did it come from? Zika has been a very obscure virus for a very long period of time. It was first isolated in 1947 in Uganda in the Zika forest. Somehow an eye got lost over time, and it was found in a rhesus monkey that had been placed as a sentinel in a cage in the Zika forest. And then it was isolated, and then shortly after that, the same virus was also isolated from a mosquito in the same forest, and this was a strong indication that it was an arbovirus transmitted by mosquitoes. Now, Zika virus symptoms are usually very mild. It is estimated that about 1 in 5 people become actually symptomatic, and if a person develops symptoms, it is a fever, often a rash, joint muscle pain, conjunctivitis. The virus course is often mild. Viremia and symptoms typically resolve within a week, and viremia is sometimes only peaking for 1 or 2 days. It has similar symptoms as dengue or chikungunya infection, which sort of complicates the initial diagnosis. And what I always discuss with my students is a very interesting paper in emerging infectious diseases. And this is a paper from 2011, before the Zika outbreak. And this provides anecdotal evidence that sexual transmission of Zika virus may occur. And this is very unusual for arboviruses, but nonetheless a very interesting case. In 2015, the year of the outbreak, it became clear that Zika could cause microcephaly in newborns and Guillain-Barré syndrome in adults, and these are more serious outcomes of Zika virus disease. So for example, microcephaly is defined as an abnormal smallness of the head. It's a congenital condition that is a result of incomplete brain development due to Zika virus infection during pregnancy. It leads to mental retardation, to deafness, behavioral problems, long-term effects on health. And these children who are born live with Zika virus infection and microcephaly, they need constant care for the rest of their lives. And I often say that although the Zika epidemic is over, it's about 10 years ago now, the effects of the Zika epidemic are still there because thousands of children were born with this syndrome and need care for the rest of their life. So it's still quite dramatic. Now, Zika virus has spread from Uganda to Brazil in about 80 or so years. It started here in 47, then it has remained silent for about 60 years until the virus surfaced in 2007 on Yap Island. This is part of Micronesia, and a lot of people here became infected with Zika virus. Then it turned up in some other islands here in New Caledonia, the Cook Islands. It made its way into Tahiti, a very pretty place. And then it turned up on Easter Island in Chile. If you look closely, you see that the statues in Chile also have microcephaly. And then the virus made its way into Brazil, where the major outbreak of Zika started. Now, at first, it was unclear how disastrous this virus infection actually was, but at the same time that the Zika epidemic was ravaging through Brazil, there was an increased incidence of babies born with smaller craniums, so microcephaly cases. And they are plotted on this graph here, and this is the sort of the epicenter of the microcephaly cases. And it took several months before this initial correlation between microcephaly cases and the actual Zika spread throughout the country was found to be a causal relationship. And now it is very clear that Zika virus indeed is the cause of this microcephaly. And it also became clear that men infected with Zika virus can carry this virus for prolonged periods of time in their semen. And that is in line with that anecdotal case that described that Zika virus could potentially be sexually transmitted as well. Now, a little bit more about the details of the virus itself. So Zika virus is a flavivirus, meaning it's a positive strand RNA virus with a capsid and an envelope carrying the envelope glycoprotein, which are present as dimers. It is internalized into a cell through receptor-mediated endocytosis, and then when the pH drops in this endosome, there's a structural conformational change in the E dimers. They form trimers, and these trimers expose the fusion peptide, and a fusion can occur, and the viral RNA escapes. Then after replication, the virus assembles at the ER membrane, and it sort of butts into the ER lumen as an immature particle. And then when it moves through the Golgi system, there furin cleavage of PRM takes place, and this makes the particle mature. And then the mature, smooth particle leaves the infected cell and then can find cells to infect again. So during infection, two types of particles are formed. First of all, there's this 50 nanometer complete particle that is comprised of a capsid and E dimers, which you can see on the surface. Here you see a scaffold of 3 dimers, so these are 6 subunits of E, all connected to each other, and it is believed that this is a TS3 triangulation number 3 particle containing 90 E dimers. But also in many flavivirus infections, there's also non-infectious particles. We call them subviral particles, SVPs. They do not contain a capsid, they do not contain genetic material, but nonetheless, they present with these E dimers on the surface, and they are really good antigens as well. And it is believed that this is a TS1 particle with 30 dimers. When the Zika virus epidemic started, we thought we should help and we should offer our expertise on VLP formation, and we wanted to build further on our success and our study on chikungunya, and we thought it was going to be quite easy. Let's just express VLPs of Zika and start a vaccine trial. So what we did is in the recombinant baculovirus, we cloned the capsid, the PRM and E proteins in order to produce these VLPs and these SVPs, and we also generated the construct without the capsid protein to only produce these SVPs. Now, this was quite successful. We were able to produce high numbers of SVPs and VLPs. We also looked at the size distribution, and if you co-express the capsid, you get a bit more of the larger particles, so more VLPs than SVPs, and if you express only PRM and E, you get more of the smaller particles. So we were very happy that we could generate these particles so easily. So then we got in contact again with Andreas Zuurbier, and he had just set up a mouse model, an IFNAR knockout mouse model that is highly susceptible to Zika virus disease. And here at week 0, we vaccinated these mice either with Zika VLPs or SVPs. We used chikungunya VLPs as a negative control or a PBS group. And we also had a positive control group, which we infected with a non-lethal dose of Zika natal. So these mice recover, and we also tested the antibody response in those mice. We gave these mice 2 boosters at week 4 and week 9, then assessed the antibody levels in the blood of these mice, and then challenged these mice with a virulent Zika strain from the outbreak. And after that, we followed up viremia. And so we were very hopeful that our vaccine would do the job. And indeed, when we look at the endpoint ELISA titer results, so this is antibody levels that are specific for Zika, we saw that our Zika VLPs and SVPs generated high levels of antibodies that were specific for Zika, although the Zika natal infection even generated higher antibody levels, and chikungunya VLPs and PBS negative controls did not generate antibodies. But when we then next looked at the neutralizing capacity of these antibodies, we were highly disappointed to find out that these antibodies did not neutralize at all. So in contrast to antibodies from the Zika natal infected mice, the Zika VLP vaccinated mice did not generate any neutralizing antibodies. So we moved on to the challenge nonetheless. And what you can see here is evenly disappointing results. You see that the control groups develop viremia at day 3 post-infection. Then viremia disappears after a while. The previously vaccinated mice with Zika natal, they did not develop viremia, they were protected, but our Zika VLPs and SVPs were very poor vaccines. And basically this disappointed so much that we left the project for a while because we had no clue how to proceed. Then we got in contact with other people and we talked about our results, and we realized that our vaccine material was Zika specific, but there was probably something wrong with the conformation of the epitopes on our particles. And one of my former PhD students had moved as a postdoc to North Carolina to the lab of Aravinda de Silva. And there they worked on panels of monoclonal antibodies, mainly for dengue, but also for Zika, just to get a better grip on what are important determinants of immunogenicity on dengue and Zika particles. And they used a specific monoclonal antibody, it's called C10. And that antibody is quite special because it is capable of binding quaternary epitopes. You can see the structure of the C10 antibody here. And so with one arm of the antibody, it binds to the dimers on the surface of this Zika particle here. And then the other arm of the antibody is across the dimers in this scaffold and binds to this. And you can imagine that if the antibody binds to this structure, that this particle has a very hard time to undergo conformational change and will probably be neutralized by this antibody. And it is therefore not a surprise that this is also a highly neutralizing antibody. So the lab in North Carolina invited us to send our vaccine material to them for their analysis on the interaction and the binding with the panels of monoclonal antibodies. And in this study, we compared the binding of the antibodies to wild-type Zika, to a Zika E subunit that does not form dimers, to a Zika VLP that we generated, and to the Zika SVP that we generated. And if you use an antibody, for example, 4G2, this is a pan-flavivirus antibody that binds to a simple epitope, then you can see that there's a good reaction to all the antigens. But however, when we start using monoclonal antibodies, and predominantly the C8 and C10 antibodies that specifically recognize quaternary epitopes on the Zika virus particle, we see that they have a very poor binding to both the VLP and to the SVP material that we produced. And so these antibodies bind much stronger to wild-type Zika, and this clearly indicated to us that there's something wrong with our VLPs and that we needed to address. Now then we thought a bit more about our expression system, and we realized that the medium that we use in the baculovirus insect cell expression system has quite a low pH. It has a pH of around 6.2. And 6.2 is quite similar to the pH in the early endosome when the virus enters a cell. And in this low pH environment, then it is known that alpha and also flaviviruses undergo structural rearrangement. So here's an example of an alpha virus spike, for example, chikungunya or Sinbis. It has trimers of E2 and E1, and upon low pH exposure, then alpha virus E2 here in yellow is displaced to expose the fusion peptide that resides in the E1 glycoprotein. Quite similarly, in flavivirus dimers in low pH conditions, they also undergo conformational change, and then they start forming trimers where the fusion peptide is also exposed upward to find the membrane it wants to fuse with. And it is thought that these trimers are not good antigens, and therefore they should be avoided in flavivirus vaccines. Because if you're capable of neutralizing dimers, then potentially virus particles cannot undergo this conformational change. And we hypothesized that the low pH in our insect cell expression system was the cause that perhaps our antigens were mostly in the trimer conformation as opposed to the dimer conformation that we wanted. So then we thought of a simple trick, just simply grow our VLPs and insect cells at pH 7, but that is not possible because then the insect cells die. So we got in contact with a biotech group in Portugal at IBET, and we had collaborated with them before. And what they did is evolution of cell lines. And what they could do is to adapt cell lines to other temperatures or other pH conditions. And we asked them whether they would help us adapting SF9 cells to a neutral pH. And that's what they did. So in steps from pH 6.5 to 6.8 to 7, they adapted these cells until they had a good growth rate. And this took about 50 to 60 passages before these cells were fully adapted to growth at a neutral pH of 7. And then we tested whether these cells could still be infected with baculovirus, and we could do so. And also these cells were still capable of expressing Zika VLPs. Now then we use these cells to express the Zika VLPs, and this was done in Portugal, and then the harvest was sent to the Netherlands, and we isolated and we purified the VLPs. Then we send them off to North Carolina for analysis using their monoclonal antibody array. And what we now see is that the C10 antibody, which does not react with VLPs produced at the low pH at 6.2, now suddenly are able to bind the VLPs that are produced at neutral pH. So we had at least solved part of the problem. But we were still not satisfied with these readings because they were still lower than the wild-type Zika. So then we read the literature a bit better, and there was an interesting amino acid substitution was described that locked the flavivirus dimers in place. And initially this mutation was developed for dengue dimers, but it also worked for Zika, and it's described in these papers over here. So basically what happens is that the alanine at position 264 is mutated into a cysteine. And because these dimers are very close to one another, then these cysteines, they can form a disulfide bridge, which covalently locks these dimers in place. And of course, you cannot do this with a live virus because then the virus would not be infectious anymore. But we're focusing on virus-like particles here, and VLPs are not infectious. So you can basically modify them any way you want. And when we did this, when we introduced this mutation, we not only had E monomers on the Western blot anymore, but also a substantial proportion of our E proteins were now in the form of dimers. And so we were very happy to see it. And this was another trick that we had to do to improve our VLP design. And so when we now compared the antibody binding to the VLPs produced at pH7 and incorporating the mutation, we can now see that these antibodies, predominantly the C10 antibody binding the quaternary epitopes, now has a much better binding to this VLP material. So we had to do two things: produce them at pH7 and introduce this mutation. Then we again purify these VLPs and send them off to Andreas Suurbier in Australia to do another vaccination study. So again, we vaccinated, and this time only with SVPs or with SVPs carrying the mutation. Both these vaccines were produced at pH7. We had a PBS group and a control. And then 3 and 7 weeks later, we gave them booster vaccinations. At week 10, we looked at neutralizing antibodies, and then we went on for a challenge and detection of viremia in the week after. And in this mouse model, we also tested the testes of these male mice 1 month after harvest because they are severely infected by Zika virus infection. And to our happiness, we now saw that in the neutralizing antibody test, we could now finally see that our VLP, or I should say SVP, carrying the mutation generated a neutralizing antibody response against Zika, which came close to neutralizing antibody levels after non-lethal infection. And that the SVP vaccine without a mutation was much less immunogenic. So we really needed this mutation to get a neutralizing antibody response. We were then confident to move on to the challenge, and here what you can see is that the control groups develop again viremia starting from day 1, and that our vaccine group was a flatliner in this assay, meaning that this vaccine protected from Zika virus-induced disease. Here you can also see that the subviral particle produced at pH7 without a mutation did somewhat do its job, but did not completely protect from viremia. So you really need this mutation in the vaccine. Now, these mice after challenge, if you look at them after about a month, then you can also see that Zika virus is infecting the testes of these mice, and then they actually start to shrink. And so this is also a nice readout for Zika virus-induced disease. And here you can see the control group, which was vaccinated with PBS. Of course, that doesn't do anything. And if you challenge them with Zika, 1 month later, these testes have become really small, and you can detect lots of testicular damage in this testis material. Both vaccines that we generated actually protected against testicular damage, although this SVP vaccine did not fully protect against viremia, but this one did. So in the end, all was well, and we had generated a successful Zika vaccine, and that is what we published in the paper. So just to sum up, what I presented to you is that we generated VLP and SVP vaccines that initially failed to protect mice because they had no good quaternary epitopes. We needed a substitution in the E domain to lock these dimers into place using a disulfide bond. And we also needed to adapt the SF9 insect cells to growth at neutral pH to produce high quality SVPs. The stabilized SVP vaccine now induced high levels of neutralizing antibodies and was able to protect the IFNAR mice against viremia and testicular damage. And so we, as a take-home message, I would say that the E dimers arranged in this conformation make sure that the quaternary epitopes are recognized by the immune system, and that this is a requisite for an effective Zika vaccine. There's of course always limitations to a study, and there's more than these here. There's a couple in the paper, but there's also 3 limitations of the study that I would like to highlight now. So first of all, the E-dimerization by the mutation that we used is not complete. So what does that mean? Zika vaccine, is that actually a good idea or not in the view of antibody-dependent enhancement, interaction with dengue? And I also have a slide on baculovirus expression of the Zika SVP vaccine and how to get rid of contaminating baculovirus particles. So first, a few words on the E-dimerization. As you can see here, is that in our vaccine with the mutation, still quite a substantial amount of the E protein is in monomeric form and probably does not contribute to the protective immune response that well. So in theory, the more dimers you make, the better vaccine you make. But whether this is really true, we do not know. We would need to investigate it. And we think that in the future we can use structure-guided design to produce more stable virus dimers, and hopefully there will be some really cool and handy AI tools coming in handy to produce such better vaccines. And we have made some good progress already with dimers of Japanese encephalitis. Then another question is, are the E-dimers alone sufficient? And in this case, I presented the E-dimers in the form of a particle in an SVP or in a VLP, but it may also be that the E-dimer by itself is already a good antigen, and this would need to be tested in future studies. So do we really need the scaffold as a virus particle, or is dimerization alone, is that enough already? Then a second point that I get a lot of questions about always, and which I still do not have a clear answer to, is what is the interaction of the Zika vaccine with dengue virus? And as you look at the phylogenetic tree of flaviviruses, then you can see that the dengue virus clade is quite close to Zika virus and Spondweni. But it is not a dengue serotype. And what is known of dengue is that if you have become infected and you recover from dengue virus infection of one serotype, you run the risk of developing more disease if you become reinfected with a subsequent serotype. So in other words, there's no cross protection between these viruses, or at least there are antibodies that are cross-reactive, but they are not protective. They are sub-neutralizing and they enhance rather than prevent disease. And the current line of thinking is that these sub-neutralizing antibodies, they bind to the heterologous serotype, fail to neutralize this virus particle, and actually through FC receptor mediated uptake, they actually promote the infection of immune cells. And it could be that a Zika virus vaccine may do the same thing with dengue, or that dengue vaccines may do the same thing with Zika. And that is, of course, a question we need to ask ourselves before we decide that vaccination against Zika should take place. Now, there's lots of papers on this question. I highlighted these two. So this is a study by the group of Eva Harris that showed that primary exposure to Zika virus is in fact linked with increased risk of symptomatic dengue virus infection, with a certain but not all serotypes. And this is another paper that shows that previous exposure to dengue virus can actually increase Zika virus infection in rhesus macaques. So if you have to ask me the question, is it a good idea to develop a Zika vaccine? I am in doubt. I think it could play a role, for example, as a traveler's vaccine, or maybe it should be given in combination with a tetravalent dengue vaccine as a sort of a 5-valent shot to protect against dengue and Zika in a single vaccine, and maybe that is the way forward. Then finally, in the beginning, I showed you that as a workhorse for our VLP production, we use the baculovirus expression system. We're a strong believer in the power of this system. It has given us the Novavax vaccine. It produces HPV vaccine, flu block. There's numerous examples of that. And today I've shown you that we can also use this to produce Zika VLPs. But there's also a downside to this system, and that is namely the co-production of baculovirus in this system. So if you produce VLPs, in the end of the day, you always have to separate your VLPs from the baculovirus material, because this is an unwanted contaminant in human vaccines, so it has to be purified away. And this, of course, is a costly business and sometimes really difficult as well. So what we have done in the past couple of years, we have now generated a new baculovirus expression system. We call it bug-free temperature sensitive or thermo switch, in which we build in a genetic thermo switch in the baculovirus, and we can switch on and off as we like the production of baculovirus in the insect cell. And for example, if you have a bioreactor with insect cells infected with the recombinant baculovirus, at a certain point, all the cells have become infected. We can decide to switch off baculovirus production by increasing the temperature in the bioreactor, and then we get no longer production of baculovirus, but the production of virus-like particles still continues. And in this way, we have shown that we can make preparations of VLPs of chikungunya, West Nile, Coxsackie, and foot and mouth disease virus with 1,000 times less baculovirus contamination. And we believe that this will facilitate downstream processing. And if you're interested in this paper, you can find it in Trends in Biotech since last week. So then I come to the final slides with the acknowledgements. I have to say lots of the work I presented today has been conducted by Sandra Abo, a former PhD student in the lab, but she had help from lots of people in our laboratory, also of the bioprocess engineering department where they did the bioreactors. We thank a lot to the people at North Carolina, Stefan Metz, Ryan, Arafinda, to think along with us what could be wrong with our VLPs. Then, of course, a big shout out to Andreas Suurbier with his animal models. Without him, we could never do vaccination studies the way I presented today. And I bet Portugal for having this really cool adaptive evolution technology to make better insect cell lines for better protein expression. And with that, I end, and then we move on to the second part of this presentation.
Thank you so much for a fantastic presentation on really exciting work. And just wanted to have you spend a few minutes talking about or thinking about how would you explain why this is important for the general public?
Yeah, so to answer your first question, Stacey, why is this study important? I think what our study has shown is that not all vaccines work for all viruses. Of course, we were very confident that we could actually do this. And given our experience with chikungunya, we had also generated VLP vaccines against Geta and Mayaro virus. It was not a problem. And then we thought we could do it with Zika, and it turned out to be really difficult. And we had to go back and understand what was wrong with these particles. And for that, we needed the collaborations. I think that is also a take-home message, that if things do not work, get help from others, steal their brains, and set up collaborations to move ahead.
Okay. Gorben, you showed also very nicely how such a project is really an international collaborative effort between different research institutes, and without it, it would never be possible.
True.
That's already indicating a little bit what that means internationally, but can you also say a little bit broader on a more personal scale what that means for family, friends, and public?
Yeah, so in this study, I worked mostly with people who I knew before from other projects, and of course everyone has its own favorite collaborators, but we also had to reach out to groups who we had not worked with that extensively before, and that worked out really well, and that has also led up to follow-up collaboration. So that has been really great also for other projects. What it means for family, friends, and public is I think that by talking about these studies, that we show that we as scientists actually care about real problems, that we're not only at academia to study things we find interesting, but that we also engage in problems in the world, and that we try to give our expertise, whatever we can give to society, and do good science, and help the science proceed. We did a similar thing in the COVID pandemic, as many other laboratories did, and I think that is really recognized also by the general public and family and friends.
Yeah, good point. And
Um,
I see a lot of young people in the Q and answer sessions,
yeah,
and they're asking all these questions. How can we contact? How can we make? Yeah, they're clearly searching for networks, and maybe it's also good a little bit for those young people to tell them a bit about careers as a scientist and how you ended up in science.
yeah. So my career has evolved from being an undergrad student to a grad student, postdoc. So the general thing. I never had to make difficult decisions. So sort of, I always flowed from one into another in a sort of natural way. But sometimes it is difficult if you have to make a choice in what direction you want to steer your career. In the end, there's not for everyone there is space for an academic career. I realize that even though as a student, if you really like to be at a university, maybe there's no spot for you how to go about that. So you always need to have a plan B, I guess. If I think about myself, how I ended up as a professor at the university, it was also a bit of luck, I guess. When I was doing my postdoc with Alex Kromick, I was really into science. I really wanted to stay in science, but I had multiple options. I could as well have gone into industry, and I think when my wife and I moved back from Australia, back to the Netherlands, all options were open, and we decided just to go for the jobs that were offered or were available. And it turned out that she went into industry, and I could pursue a career as an academic. But
Yeah.
flipping the coin differently, it could have been the reverse. And yeah, so you cannot always decide which direction you want to go. There's also luck involved, and
Yeah.
yeah.
Yeah, so in view of time, maybe we should move to content questions right now. I've collected some questions, and unfortunately, we can't answer all questions live, but we will try to answer the other questions afterwards. So I
Yep.
selected a few. One of the questions which I thought was relevant was on how quickly actually Zika is mutating in real life, and how difficult is it to keep up with the vaccine design.
Oh, I see. Yeah, so that question is probably coming forth from the COVID pandemic, where that is really relevant, right? With all the different
Yeah, I
variants.
guess, yeah.
Yeah. For Zika, my expectation is that Zika is a single serotype, which means that probably a vaccine against one strain will also confer good protection against another. If you really want to answer this, you of course have to test it. We do see differences between Zika strains, for example. If you use an African clade strain and use it in a mouse model, it is quite different in terms of disease that you see with the Asian outbreak strain that circulated in South America. But still, I think in terms of an antibody response, I still think that the protection is almost equal, and that we do not need to worry so much about mutation of Zika.
Yeah.
Perhaps also keep in mind that Zika always travels back and forth between humans and mosquitoes, so it cannot unlimited mutate, because there's evolutionary limits to the mutational space in mosquitoes and humans.
Yeah, there were also a few questions about stability of the VLPs, and that's particularly relevant, I think, if you want to use this vaccine in the tropical areas.
Yep. Yeah, so it is a great question, and we do not have a good answer to that. The VLPs are, of course, enveloped, and by definition, then they are less stable than, I would say, a VLP that is a protein only. For example, if you have a human papillomavirus, VLP is much more stable than this one. What we saw in our study is that we also did an initial experiment, which I did not show today, but it's in the paper, where we tested the vaccine with and without adjuvant. And in one of the experiments, we actually have indications that adding the adjuvant destabilizes the vaccine. And so if we think about that, I think it would be worthwhile to study the stability of the VLPs in the presence or absence of adjuvants in the future. And maybe there's a lot of improvements to be made in choosing a different adjuvant.
Okay, then there was also a question where somebody did not really understand what the difference was between the SVPs and the VLPs.
Okay, yes. Yeah, that SVP is an abbreviation of subviral particle. I had a slide on it in the beginning. You can always look it back, but that is basically a particle that is co-produced during a virus infection that contains no capsid and no genetic material. So it's basically an empty vesicle that has dimers on the surface, and that is what an SVP is. And they're highly immunogenic, although of course, the E dimers are arranged in a slightly different way on the surface.
Okay, and maybe a bit related to this is also somebody was thinking, well, we have these different type of arboviruses and these different types of flaviviruses. So has anybody ever tried to make a multivalent VLP vaccine for Zika, dengue, and Japanese encephalitis, for instance?
I cannot recall that people have tried it. It could be a good idea to make a combination vaccine, and I would especially then choose viruses that are transmitted by the same mosquito and circulate in the same area, because I think in the Netherlands we may benefit from a West Nile vaccine, but not so much from a chikungunya vaccine, because they're transmitted by different vectors. So, for example, if you think about Brazil, maybe a Mayaro chikungunya Zika combination vaccine could be a nice idea. But we have not tried it, but I see no reason why it could not be done.
Yeah, and somebody also had a question about vaccines, how this VLP candidate vaccine, I should say, compares to the existing Japanese encephalitis vaccines and dengue vaccines. How are these produced and how are these
Yes,
made, and what's
yeah,
better?
I cannot give an answer to that. We have not compared it. So for Japanese encephalitis, there's different vaccines around. I think in Asia and Australia they use a live attenuated, probably giving a pretty long immunity. Here in the Netherlands, we also have a Japanese encephalitis vaccine, but that's a killed virus vaccine, and you need multiple boosters. And I would assume that a VLP-based vaccine is in that sense a bit similar to a killed virus vaccine. You need multiple boosters to keep the antibody levels up, would be my guess.
And there's actually another interesting question just came in. You described about this antibody-dependent enhancement, with let's say that you see with that you might be afraid of if you use Zika for dengue, but is it also working the other way around?
Yeah, so we have not studied it, but the two papers I showed is both ways around. So in one study, then prior Zika infection has an effect on more symptomatic dengue, and in the other study, then dengue virus infection first makes Zika virus disease worse, and that was in a macaque model.
Okay. And apparently some people did not really understand what you were doing in your studies with the neutralizing antibodies, because a few people thought you were using those as well as an immunization strategy, but that was not the case, was it?
No, no, that was not the case. So we in the study, and this was done by the collaborators in North Carolina. So they had a panel of monoclonal antibodies, and they used this in ELISA-based setups just to study the binding efficiency of these monoclonal antibodies to antigens. So we have all these vaccine candidates, and then by using these monoclonal antibodies that recognize epitopes that are important for protection, we can fish out those vaccine designs that have the epitopes presented in the conformation we want.
Yeah, so that you have the highest chance of an effective
Yes.
vaccine in the end.
Yeah,
Yeah.
exactly. That's the idea.
Yeah, then for maybe a final question, somebody said, did you try to make the VLPs also in mammalian cells? And what would be the pros and cons of both systems?
It's a very nice question. Of course, that is another strategy, and I think one of the benefits is that you do not have the pH issue, because the pH of mammalian cell culture medium is more close to neutral pH. So I do not think that issue is there. For large scale production, it may be less competitive than the baculovirus expression system, because you would have to do large scale transfections of, for example, HEC 293 cells in suspension. I know it can be done because for chikungunya, the VLP vaccine that is now being produced by Bavarian Nordic is made in a similar way. So I think it can be done in a cost-effective way. I think there are simply two systems that can both be used, and glycosylation will be a bit different.
Yeah, that's what the person is also asking about. Yeah.
Yeah, so in insect cells, generally the glycosylation of the envelope glycoproteins is a bit more predictable. In insect cells, you could say a bit more boring, because you know what you know what you get. In mammalian cells, it's always the question: how are the glycans trimmed? How are they modified? This can vary greatly from cell to cell. It can also greatly from glycosylation site to another site, and if you really want to know this, it can be determined by mass spec in great detail what the glycans look like.
And with
Okay, I
that,
think
I
in
have
view
to,
of time, we give it back here to
yeah,
Stacy.
yeah,
Thank you very
no,
much, Gorben. Sorry.
thank you. Gorben, can you go forward a couple slides? So to thank just outstanding presentation today by Gorben, moderating by Monique, all of you for participating in today's webinar, and of course the wonderful ASM staff. We could not do this without you. Next slide, please. Think about JV when you're ready to submit your manuscript. Go online, read Gorben's paper. We are now open access, so it's freely available to everybody. Next slide, please. And we hope that you will join us for our next seminar and webinar, where we're going to talk about giant viruses in amoeba. So something very different. There's information about that, and you can contact us for more, for the links. And final slide, please. And we always want your feedback. It's very important to us, and there's ways you can get in touch with us. either by emailing us at jbieic on Blue Sky or directly to jb@asmusa.org. So with that, we're going to close out this session and thank all of you and have a good rest of your day wherever you are in the world.